Hi, today we are going to talk about genetic recombination in bacteria. Genetic recombination refers to the exchange between two DNA molecules. It results in new combinations of genes on the chromosome. But how did the chromosome segment get into the cell? The answer is genetic transfer. Genetic transfer is the mechanism by which DNA is transferred from a donor to a recipient. In bacteria, genetic transfer can happen three ways. The first way is by transformation, the second by conjugation, and the third by transduction. But today we are going to talk about the process of transduction. Transduction is the transfer of genetic information from a donor to a recipient by the way of a bacteriophage. Bacteriophage are viruses that infect bacterial cells and use them as hosts to make more viruses. Transduction can either be specialized or generalized transduction. But before we discuss that, you should know first the bacteriophage life cycle. Talking about viral replication, we already know that viruses are made of only proteins and one type of nucleic acid, which means they have no organelles to make copies of themselves with, and they have to get inside other cells in order to use their ATP and their organelles. So the first step is to get inside the cell. Remember, there are three ways of getting into a cell. One of the ways of getting in is that the bacteriophages inject their genetic material inside of the cell. Whereas every other type of virus uses one of two ways. If they're non-enveloped, they trick the cell's receptors into letting them in. Or if they're enveloped, they can either use that receptor trick or they directly fuse with the membrane. And once that genetic material is inside of the cell, the virus has hacked in and it's ready to do a takeover. This is where the virus makes a choice. The virus is either impatient and takes over right away, or is patient and bides its time. With the first choice, the impatient virus goes ahead and takes over the cell's machinery. It takes over their ATP, ribosomes, nucleic acids, amino acids, and starts making copies of the virus's genetic material, which can be RNA or DNA. And also, it takes over the virus's proteins to make their protein coat, and these will self-assemble. That means they just come together on their own to form fully functional viruses, and because it keeps making more and more of these, it will eventually force the cell to lyse or break open, and once it breaks open, all of these viruses are released into the environment. And if there are other cells nearby, then this army can start marching out to infect and hack those nearby cells and create more armies. With the other option, that is where the virus decides it's just going to sneak in and hitch a ride. It thinks that the bacteria seems to be doing fine on its own. And maybe there aren't any other hosts nearby, so there's no reason to kill off the host, because lysing it would kill the host, and we don't want that. We're just going to keep the host cell alive. And in order to sneak in and let the bacteria do its thing while it's waiting, it's going to combine with the host's genetic information so that the host really can't tell that it's there. It's basically quietly sitting there because it's repressed. There are repressor genes on this virus, so it's not expressed or transcribed, and it cannot do anything. And this is called a provirus, or you might also hear the word prophage. And again, because it's not doing anything, this is called a dormant or latent phase. So the bacteria just does its own thing. It continues replicating, and of course the virus is already there. So it will replicate when the bacteria does, because it's part of its genome. But about 1 in 10,000 times that this happens, or if something happens like the bacteria is exposed to UV light or something, there's something that weakens the repressor gene that we talked about on the virus, so it's no longer quiet. And the host's genome, like most other genomes, will start wanting to repair itself. That means it will cut out to try to repair its genome. It will excise out part of its DNA that just happens to be the virus. And now, the virus is active. It's ready to make copies of itself, lyse the cell, and get its army out into infecting other cells. So the official terms for the impatient method is the lytic cycle, and hitch-a-ride method is called the lysogenic cycle. So now you should have a good idea of what the lytic and lysogenic cycles are in viral replication.
Transduction can either be specialized or generalized transduction. General transduction is transduction in which potentially any bacterial gene from the donor can be transferred to the recipient.
The process of generalized transduction is transduction in which potentially all bacterial gene from the donor can be transferred to the recipient. The process begins with the bacterial host cell, also known as the donor cell, and gets infected by the phage. Then, the host cell DNA is broken down into smaller pieces. Proteins and phage DNA is also synthesized. Next, the bacterial host DNA is packaged in one of the viral capsids that are released through lysis of the bacterial cell. The next step is where the transducing phage with host DNA infests new recipient cell. Recombination also occurs here. Then, the recombinant new cell has a mixture of the donor DNA and one of its own DNA. Its genotype is different. In specialized transduction, on the other hand, only certain bacterial genes can be transferred. These genes, as you will see, must exist on either side of the prophage. Specialized transduction requires a phage that uses the lysogenic cycle for reproduction.
The process of specialized transduction occurs when the prophage DNA integrates into the bacterial DNA. Then, the prophage DNA cuts incorrectly and exchanges its DNA to that of bacterial host cell. The phage capsids contain bacterial host DNA, which is transferred to a new bacterial recipient cell. The transduced phage is transferred to a new cell and recombination occurs. The recombinant cell DNA has a different genotype, which is different from the donor or recipient cell genotype.